We have to keep in mind that there's also cis transition factors, as well as regulatory factors. So what this does is, this could account for the genetic differences between the groups. But at the same time, NRF2 is part of a pathway of a genetic domain called BZIP, which stands for Basic Leucine Zipper. So essentially what these things do is they help proteins attach to DNA. So for the BZIP, the most basic one is 2 alpha helices, like that. And for NRF2, I tried to look at the crystallography. Unfortunately, PC's neuroscience program doesn't have to make you take chemistry, so it looked like this. But the main takeaway is that it is specialized for reducing the antioxidant response element. But this was not enough in order to determine full homology. What they also needed to do was, there is a protein called SKIN1 that is found in Drosophila, and it forms a homodimer. And it has different developmental implications rather than NRF2, which is reducing oxidative stress. So even though, despite that the genetics themselves were similar, their homology doesn't necessarily mean that it's just like, if they look structurally similar, they can also have different functionalities. So the last thing they did was they took, over here, they did assays to see if NRF2 and KEAP1 were together and bound, and they were tightly bound. So when they did them together in a gel, they found that they were conserved so that it would appear on a Western blot. Despite having such high homology and that it is actually affecting the target, at the same time, it has low affinity. So that means there's different off targets that occur. So the ones that we're going to be talking about today is the liver, close enough, the kidney, and I was going to draw something phallic, but it's just the reproductive system. Okay. So for the liver, what happens is, they did a test and they would do liver brain weight ratios, so milligrams per kilograms per day. And it didn't matter which animal they used. It found that no matter how much you gave, it would not change the functionality of the liver. However, what they did find was multifocal hepatic necrosis, which means that spots come on the liver. And they also found something called bile duct hyperplasia. So that means this part right here would enlarge. But if you stop taking Tecfidera, some of these spots and tissue damage would eventually go away, but this enlargement would also go back down. So yeah, and then
And so in addition to the liver toxicity, we're also going to speak about the reproduction system. So there were 2 different, well, I'm going to speak about male and female. They looked at the infertility in males and also the embryonic development and postnatal development in rats. So in the males, 2 important things were seen. One, it's that, there were histopathological changes in the testes, and the first one is testicular Leydig cell hyperplasia. That.
The Leydig cells are one of the cells types in the testes, so that would make.
Yep. So it's basically the inability of your testes to produce testosterone or other sex hormones, and it could lead to underdeveloped or partially developed genitalia, and it could also lead to infertility. So in that same study, they looked at dogs, and they found the degeneration of the seminiferous
Seminiferous tubules.
Yes, and that's basically where the creation of sperm is. So even though they found all of these conflicts, they said that it doesn't affect the motility nor the count of sperm in the animals. So then, we're gonna look at the females. And in the females, they kind of did, it's called, I named it a Noel, is that how you pronounce it? So it's basically no observed adverse effect level. So it's basically just comparing the highest dose that they could give to the animal of Tecfidera until there's obviously a very high adverse reaction. And an example of this is that they found that in females that are pregnant, the NOEL is 20 milligrams per kilograms. And they also found that in very high doses of DMF, the pups are there's a reduced weight in the pups. There's also alterations, which it's one of the alterations, it's that it's a very slow process of the ossification in the metatarsals. And then there's also they looked at the female rabbits.
So their bones in their feet just don't harden?
Yeah. So also in some of the females, they found 4 different miscarriages. And in addition to that, they found that once the pup was born, there was a very a delay in sexual maturity in the male pups. And regardless of all of these things that they found, they say that there were no deaths while taking Tecfidera. So I mean, they're obviously not recommending that women who are pregnant to take Tecfidera, but they're just kind of showing that it's not gonna be a bad effect in the fetus.
Okay, so then lastly we looked at like kidney toxicity and how Tecfidera affects the kidneys. So they did studies in rats, mice, monkeys, and dogs. And I'm just gonna tell you a little bit about the rats and the monkeys. So basically all different animals, they found different results in the kidneys, so they all affected it differently. But in the rats, they did a repeat dose study, which is just a study that is generally done to like determine toxicity, and they repeated the dosage daily. And they found that this led to neurotoxicity, which is just a damage of the kidneys, and it causes you to not be able to like eliminate your urine and your waste properly. And then in the monkeys, they had a low dose and a high dose. So the low dose was 25 milligrams per kilogram, like body weight, per day. And the high dose was 75 milligrams per kilogram per day. And they found that kidney alterations correspond to single cell necrosis, and that the regeneration of the cortical tubular epithelial cells, but that was more severe in the high dose, so the low dose wasn't as bad. And then they also found that in males of the high dose, there was mild to moderate renal fibrosis. And so although they found all these toxicities in the kidneys in these different species, they don't affect the safety of the human treatment dosage. So the humans are okay. And we're gonna go ahead and talk about the business side and how, although Tecfidera has off-target implications, it's still an important drug for MS.
So compared to the other drugs out there for MS, how do these kind of side effects and tox effects come across? They're less significant than other drugs, or more significant?
From the first presentation that we gave, it seemed to have less symptoms than other drugs.
And I did see a research data said that Gilenya had really bad heart conditions, and Albagio had really bad GI issues compared to Tecfidera. So although we don't have a Susan, at least we have 7 different patents, and those patents can be divided between the 412 milligram patents and the use patents. So I'll go into the use patents first. So our grandfather, the original patent was 2003. Was approved in 2003 and will expire in 2019. This grandfather patent just gives us the use of dialkyl fumarates, which includes DMF. It also gives us the indication, which is that we're using DMF for MS. And in addition to that, it gives us the formulation of Tecfidera. So the following patents are kind of a division of the grandfather patent. They're kind of being more specific. And so this one expires in 2019, and this one's the preparation of DMF as a microtablet. And then the use is just specifying that we're using DMF in MS. And then the following ones are also use. This one's specifying that DMF, and another one which is called methyl hydrogen fumarate will be used for MS. And then the next one specifies that we're going to be using DMF for MS, and in addition to MS, we're going to be using it for an autoimmune form of polyarthritis. And the last one, which is kind of a division of this one right here, expires in 2018, but it's a method of treatment, and it's basically saying that we're going to be treating MS through an oral form, and that oral form will come in contact with the blood peripheral monocytes, and those will come in contact with the MHF. And then the other group, which is the 412 milligram patents, although there's just one patent for this one, it's actually the most important because it's the one that extends to 2028. And this one is just basically specifying that we're going to be using DMF and MMF, which is the metabolite of DMF, for Tecfidera. And it's also saying that no other company can use an excipient form for DMF and MMF. And in addition to that, we have, Tecfidera has market exclusivity until 2018.
And that's given by the FDA?
Yes.
Okay.
And then I just kind of wanted to mention that because early this year, the European Court revoked one of Tecfidera's kind of the most important patent, and it's the US equivalent of this patent, because another company came out and said that they filed for a similar drug as Tecfidera. And the Biogen is currently appealing this, and although it might, they don't know yet if it's going to be revoked or not, but this could give them at least 4 more years with Tecfidera in Europe. And although there are no generics kind of being produced, I read an article that it said that they wanted to combine Tecfidera with a different substance for MS. And in addition to that, Biogen is also producing, it has studies for two different MS drugs. And one of those drugs they already filed for approval, and that's a monoclonal antibody. And although, as we can see, these patents are obviously going to expire pretty soon, Tecfidera is expecting to make about $4 billion in 2018, in addition to the about $196 million they made in 2013.
So they're going from 20 million to 4 billion?
Well, they're going to 4 billion, and in the first half of the sales in 2013, they made 196 million.
That's not bad. I still have stock in Biogen. You done? Anybody have questions? Good job.
Yes.
When you're talking about the reproductive side effects, were you talking about when people take, like when a woman takes it when she's pregnant, or is this a woman who has taken it before becoming pregnant?
Oh,
and
it
doesn't
would
take
be,
then?
in the studies, there were bats who were pregnant, not before.
Okay.
For the patent that was filed, that was possibly filed in Britain, was that filed in the US? Or was that?
I don't think so, it was just filed in Britain, well, in Europe
Yeah.
overall, yeah.
Sometimes, yeah, you have to file separately, and that's
Yeah.
part of how expensive some of this stuff can actually be, having to do that.
I think it's Fine Pharma, which I'm not sure if that's like a US.
What's the name of it?
Fine Pharma,
I don't know.
so it's probably European.
Well, a lot of like these companies, like Sanofi's a French company, like a lot of the companies you think about, you know, having offices here aren't American companies. Even Biogen was started in Geneva.
Anybody else?